सो हाय गाइस, दिस इज़ धीरज कुरैन। ये वीडियो है एनसीईआरटी लाइन बाय ऑडियो बुक। आप पेन लो, बुक लो, सोचो कि दुनिया ख़त्म हो गई है, तुम और बुक बस ये लो पेन, और जो मैं बोल रहा हूँ उसको साथ मार्क करते करते जाओ एंड यू विल रियलाइज़ कि एनसीईआरटी ख़त्म भी हो गया और तुम्हारे दिमाग में घुसने भी लगेगा। और जब भी दिमाग में कुछ उल्टा सीधा आए तो सोचो, उस फोकस तुम्हें अपने लाइन पे ले जाना है कि बोल क्या रहा है, इससे रिलेटेड क्वेश्चन पूछने तुम्हें, बस गो, दैट्स इट। हाय गाइस, दिस इज़ धीरज कुर्रे और आज के इस वीडियो में हम करने वाले हैं क्लास 11th बायोलॉजी का चैप्टर नंबर 10 विच इज़ सेल साइकिल एंड सेल डिवीजन। तो आज के इस वीडियो में हम इस चैप्टर का पूरा एनसीईआरटी लाइन बाय लाइन पढ़ेंगे। आप इसे रिवीजन के तौर पर देख सकते हो या फिर आप इसे बार बार रिवाइज करने के लिए फिर पढ़ने के लिए इसका यूज़ कर सकते हो। आप इसका 2x में चला के देखें या फिर अपने रिवीजन पर्पज़ के लिए अपने हिसाब से यूज़ करें। सेल साइकिल एंड सेल डिवीजन। Are you aware that all organisms, even the largest, start their life from a single cell? You may wonder how a single cell then goes on to form such large organisms. Growth and reproduction are characteristics of cells. Indeed, of all living organisms, all cells reproduce by dividing into two, with each parental cell giving rise to two daughter cells. Each time they divide, these newly formed daughter cells can themselves grow and divide, giving rise to a new cell population that is formed by the growth and division of a single parental cell and its progeny. In other words, such cycles of growth and division allow a single cell to form a structure consisting of millions of cells. 10.1 Cell cycle. Cell division is a very important process in all living organisms. During the division of a cell, DNA replication and cell growth also takes place. All these processes, that is cell division, DNA replication and cell growth, hence have to take place in coordinated way to ensure correct division and formation of progeny. Cells containing intact genomes, the sequence of events by which cell duplicates its genome, synthesis the other constituent of the cell and eventually divides into two daughter cells is termed cell cycle. Although cell growth in terms of cytoplasmic increase is a continuous process, DNA synthesis occurs only during one specific stage in the cell cycle. The replicated chromosomes DNA are often distributed to daughter nuclei by a complex series of events during cell division. These events are themselves under genetic control. 10.1.1 Phases of cell cycle. A typical eukaryotic cell is illustrated by human cells in culture. These cells divide once in approximately every 24 hours. Figure 10.1. Let's say figure 10.1. A diagrammatic view of cell cycle indicating formation of two cells from one cell. Here is G1S, G2, Prophase, Metaphase, Anaphase, Telophase, Cytokinesis, which comes under Mphase. However, this duration of cell cycle can vary from organism to organism and also from cell type to cell type. Yeast, for example, can progress throughout the cell cycle in only about 90 minutes. The cell cycle is divided into two basic phases: interphase, M phase, which is mitosis phase. The M phase represents the phase when the actual cell division or mitosis occurs, and the interphase represents the phase between two successive M phase. It is significant to note that in the 24-hour average duration of the cell cycle of human cell division proper lasts for only about an hour. Interphase lasts more than 95% of the duration of the cell cycle. The M phase starts with the nuclear division, corresponding to the separation of daughter chromosomes, karyokinesis, and usually ends with division of cytoplasm, cytokinesis. The interphase, though called the resting phase, is the time during which the cell is preparing for division by undergoing both cell growth and DNA replication in an orderly manner. The interphase is divided into three further phases: G1 phase, gap 1, S phase, synthesis phase, G2 phase, gap 2. G1 phase corresponds to the interval between mitosis and initiation of DNA replication. During G1 phase, the cell is metabolically active and continuously grows but does not replicate its DNA. S or synthesis phase marks the period during which DNA synthesis or replication takes place. During this time, the amount of DNA per cell doubles. If the initial amount of DNA is denoted as 2C, then it is increased to 4C. However, there is no increase in the chromosome number. If the cell has had haploid or 2n number of chromosomes at G1, even after S phase the number of chromosomes remain the same, that is 2n. In animal cells, during S phase, DNA replication begins in the nucleus and the centriole duplicates in the cytoplasm. During the G2 phase, proteins are synthesized in preparation for mitosis, while cell growth continues. Let's read the brown box. How do plants and animals continue to grow all their leaves or their lives? Do all cells in a plant divide all the time? Do you think all the cells continue to divide in all plants and animals? Can you tell the name and the location of the tissues having cells that divide all their life in higher plants? Do animals have similar meristematic tissues? Here is yellow box. You have studied mitosis in onion root tip cells. It has 16 chromosomes in each cell. Can you tell how many chromosomes will the cell have at G1 phase, after S phase, and after M phase? Also, what will be the DNA content of the cells at G1, after S, at J2 if the content after M phase is 2C? Some cells in the adult animal do not appear to exhibit division. Example, heart cell, and many other cells divide only occasionally, as needed to replace cells that have been lost because of injury or cell death. These cells that do not further exit G1 phase to enter an active stage called quiescent stage G0 of the cell cycle. Cells in this stage remain metabolically active but no longer proliferate unless called on to do so, depending on the requirement of the organism. In animals, mitotic cell division is the only seen in diploid somatic cells. However, there are few exceptions to this where haploid cells divide by mitosis. For example, male honeybees. Against this, the plant cells show mitotic division in both haploid and diploid cells. From your recollection of examples, alternation of generation in plants, chapter 3, identify plant species and stages at which mitosis is seen in haploid cells. 10.2 M phase. This is the most dramatic period of the cell cycle, involving a major reorganization of virtually all components of the cell. Since the number of chromosomes in the parent and progeny cells is the same, it is also called as equational division. Though for convenience, mitosis has been divided into 4 stages of nuclear division. Karyokinesis. It is very essential to understand that cell division is a progressive process and very clear-cut lines cannot be drawn between various stages. Karyokinesis involves the following 4 stages: Prophase, Metaphase, Anaphase, Telophase. Prophase, which is the first stage of karyokinesis of mitosis, follows the S and G2 phase of interphase. In the S and G2 phase, the new DNA molecules formed are not distinct but interlinked. Prophase is marked by the initiation of condensation of chromosome material. The chromosome material becomes untangled during the process of chromatin condensation. The centrosome, which had undergone duplication during S phase of interphase, now begins to move toward opposite poles of the cell. The completion of prophase can thus be marked by the following characteristics events. Chromosomes material condenses to form compact mitotic chromosomes. Chromosomes are seen to be composed of 2 chromatids attached together at the centromer. Centromers, which had undergone duplication during interphase, began to move towards opposite poles of cell. Each centromer radiates out microtubules called esters. The 2 esters together with spindle fibers forms mitotic apparatus. Cells at the end of the prophase, when viewed under the microscope, do not show Golgi complexes, endoplasmic reticulum, nucleolus, and the nuclear envelope. 10.2.2 Metaphase. The complete disintegration of the nuclear envelope marks the start of the second phase of mitosis. Hence, the chromosomes are spread throughout the cytoplasm of the cell. By this stage, condensation of chromosomes is completed and then can be observed clearly under the microscope. This then is the stage at which morphology of chromosome is most easily studied. At this stage, metaphase chromosome is made up of two sister chromatids, which are held together by the centromer. Figure 10.2b. Metaphase. Small disc-shaped structures at the surface of the centromer are called kinetochores. This structure served as the site of attachment of spindle fibers formed by the spindle fibers to the chromosomes that are moved into position at the center of the cell. Hence, the metaphase is characterized by all the chromosomes coming to lie at the equator, with one chromatid of each chromosome connected by its kinetochore to spindle fibers from one pole and its sister chromatid connected by its kinetochore to spindle fibers from the opposite pole. Figure 10.2b. The plane of alignment of the chromosomes at metaphase is referred to as the metaphase plate. The key features of metaphase are spindle fibers attached to kinetochores of chromosomes. Chromosomes are moved to spindle equator and get aligned along metaphase through spindle fibers to both poles. 10.2.3 Anaphase. At the onset of anaphase, each chromosome arranged at the metaphase plate is split simultaneously, and the two daughter chromatids, now referred to as daughter chromosomes of the future daughter nuclei, begin their migration towards the two opposite poles. As each chromosome moves away from the equatorial plate, the centromer of each chromosome remains directed towards the pole and hence at the leading edge, with the arms of the chromosome trailing behind. Figure 10.2c. Thus, anaphase stage is characterized by the following key events: centromers split and chromatid separate. Chromatids move to opposite poles. 10.2.4 Telophase. At the beginning of the final stage of karyokinesis, that is telophase, the chromosomes that have reached their respective poles decondense and lose their individuality. The individual chromosomes can no longer be seen, and each set of chromatin material tends to collect at each of the two poles. Figure 10.2d. This is the stage which shows the following key events. Chromosomes cluster at opposite spindle poles and their identity is lost as discrete elements. Nuclear envelope develops around the chromosomes cluster at each pole, forming two daughter nuclei. Nucleolus, Golgi complex and ER reform. 10.2.5 Cytokinesis. Mitosis accomplishes not only the segregation of duplicated chromosomes into daughter nuclei, karyokinesis, but cell itself is divided into two daughter cells by the separation of cytoplasm called cytokinesis, at the end of which cell division gets completed. Figure 10.2e. In an animal cell, this is achieved by the appearance of a furrow in the plasma membrane. The furrow gradually deepens and ultimately joins in the center, dividing the cell cytoplasm into two. Plant cells, however, are enclosed by a relatively inextensible cell wall. Therefore, they undergo cytokinesis by a different mechanism. In plant cell, wall formation starts in the center of the cell and grows outward to meet the existing lateral walls. The formation of the new cell wall begins with the formation of a simple precursor called the cell plate that represents the middle lamella between the walls of two adjacent cells. At the time of cytoplasmic division, organelles like mitochondria and plastids get distributed between the two daughter cells. In some organisms, karyokinesis is not followed by cytokinesis, as a result of which multinucleated condition arises, leading to formation of syncytium. Example: liquid endosperm in coconut. 10.3 Significance of mitosis. Mitosis, or the equational division, is usually restricted to diploid cells only. However, in some lower plants and in some social insects, haploid cells also divide by mitosis. It is very essential to understand the significance of this division in the life of an organism. Are you aware of some examples where you have studied about haploid and diploid insects? Mitosis usually results in the production of diploid daughter cells with identical genetic complement. The growth of multicellular organisms is due to mitosis. Cell growth results in the disturbing the ratio between the nucleus and the cytoplasm. It therefore becomes essential for the cell to divide to restore the nucleocytoplasmic ratio. A very significant contribution of mitosis is cell repair. The cells of the upper layer of the epidermis, cells of the lining of the gut, and blood cells are being constantly replaced. Mitotic divisions in the meristematic tissues, the apical and the lateral cambium, result in the continuous growth of plants throughout their life. 10.4 Meiosis. The production of offspring by sexual reproduction includes the fusion of two gametes, each with a complete haploid set of chromosomes. Gametes are formed from specialized diploid cells. This specialized kind of cell division that reduces the chromosome number by half results in the production of haploid daughter cells. This kind of division is called meiosis. Meiosis ensures the production of haploid phase in the life cycle of sexually reproducing organisms, where fertilization restores the diploid phase. We come across meiosis during gametogenesis in plants and animals. This leads to the formation of haploid gametes. The key features of meiosis are as follows. Meiosis involves two sequential cycles of a nuclear and cell division called Meiosis I and Meiosis II, but only a single cell of DNA replication. Second, meiosis is initiated after the parental chromosomes have been replicated to produce identical sister chromatid at S phase. Third, meiosis involves pairing of homologous chromosomes and recombination between non-sister chromatid of homologous chromosomes. 4. Haploid cells are formed at the end of meiosis II. Meiotic events can be grouped under following phases: meiosis I, Prophase I, Metaphase I, Anaphase I, Telophase I. Meiosis II, Prophase II, Metaphase II, Anaphase II, Telophase II. 10.4.1 Meiosis I. Prophase I. Prophase of the first meiotic division is typically longer and more complex when compared to prophase of mitosis. It has been further subdivided into following 5 phases based on chromosomal behavior, that is lactotin, zygotin, pakitin, diplotin, and dikinesis. During lactotin stage, the chromosomes become gradually visible under light microscope. The compaction of chromosomes continues throughout lactotin. This is followed by the second stage of prophase 1 called zygotin. During the stage of chromosomes start pairing together, and this process of association is called synapses. Such paired chromosomes are called homologous chromosomes. Electron micrographs of this stage indicate the chromosome synapses is accompanied by the formation of complex structure called synaptonemal complex. The complex formed by a pair of synapsed homologous chromosome is called bivalent or a tetrad. However, these are more clearly visible at the next stage. The first 2 stages of prophase 1 are relatively short-lived compared to the next stage, that is pakitin. During this stage, the 4 chromatid of each bivalent chromosomes becomes distinct and clearly appears as tetrads. This stage is characterized by the appearance of recombination nodules, the sites at which crossing over occurs between non-sister chromatid of the homologous chromosomes. Crossing over is the exchange of genetic material between 2 homologous chromosomes. Crossing over is also an enzyme-mediated process, and the enzyme involved is called recombinase. Crossing over leads to recombination of genetic material on the 2 chromosomes. Recombination between homologous chromosomes is completed by the end of pakitin. Leaving the chromosomes is linked at the site of crossing over. The beginning of diplotin is recombinized by the dissolution of the synaptonemal complex and the tendency of the recombinant homologous chromosomes of the bivalents to separate from each other except at the sites of crossovers. These egg-shaped structures are called chiasmata. In oocytes of some vertebrates, diplotin can last for months or years. The final stage of meiotic prophase 1 is dikinesis. This is marked by the termination of chiasmata. During this phase, the chromosomes are fully condensed and the meiotic spindle is assembled to prepare the homologous chromosomes for separation. By the end of dikinesis, the nucleus disappears and the nuclear envelope also breaks down. Dikinesis represents the transition to metaphase. Metaphase 1, the bivalent chromosome align on the equatorial plate. 10.3, figure 10.3. Stages of meiosis 1, prophase 1, metaphase 1, anaphase 1, telophase 1. The microtubules from the opposite poles of the spindle attach to the kinetochore of homologous chromosome. Anaphase 1, the homologous chromosomes separate while sister chromatids remain associated at their centromeres. Figure 10.3. Telophase 1, the nuclear membrane and nucleus reappear. Cytokinesis follows, and this is called as dyad of cells. Figure 10.3. Telophase 1, the nuclear membrane and nucleolus reappear. Cytokinesis follows, and this is called a dyad of cells. Figure 10.3. Although in many cases of the chromosomes do undergo some dispersion and they do not reach the extremely extended state of the interphase nucleus, the stage between the two meiotic division is called interkinesis and is generally short-lived. There is no replication of DNA during interkinesis. Interkinesis is followed by the prophase second, a much simpler prophase than prophase 1. 10.4.2 Meiosis II. Prophase 2, Meiosis II is initiated immediately after cytokinesis, usually before the chromosomes have fully elongated. In contrast to Meiosis 1, Meiosis II resembles a normal mitosis. The nuclear membranes disappears by the end of Prophase 2, figure 10.4. The chromosomes again become compact. Metaphase second, at this stage the chromosomes align at the equator and the microtubules from the opposite poles of the spindle get attached to the kinetochores, figure 10.4, of sister chromatids. Anaphase 2, it begins with simultaneous splitting of the centromere of each chromosome, which was holding the sister chromatid together, allowing them to move toward opposite poles of the cell, figure 10.4, by the sorting of microtubules attached to kinetochores. Telophase 2, meiosis ends with telophase second, in which two groups of chromosomes once again get enclosed by a nuclear envelope. Cytokinesis follows, resulting in the formation of tetrad of cells, that is 4 haploid daughter cells, figure 10.4. 10.5 Significance of meiosis. Meiosis is the mechanism by which conservation of a specific chromosome number of each species is achieved across generation in sexually reproducing organisms. Even though process per paradoxically results in the reduction of chromosome number by half, it also increases the genetic variability in the population of organisms from one generation to the next. Variations are very important for the process of evolution. Prophase 2, Metaphase 2, Anaphase 2, Telophase 2. According to the cell theory, cells arise from pre-existing cells. The process by which this occurs is called cell division. Any sexually reproducing organism starts its life from a single cell zygote. Cell division does not stop with the formation of mature organisms but continues throughout its life cycle. The stages through which a cell passes from one division to the next is called the cell cycle. The cell cycle is divided into two phases called first interphase, a period of preparation of cell first interphase, a period of preparation for cell division, and second mitosis, the actual period of cell division. Interphase is further subdivided into G1, S and G2. G1 phase is the period when the cell grows and carries out normal metabolism. Most of the organelle duplication also occurs during the S phase. During this phase, S phase marks the phase of DNA replication and chromosome duplication. G2 phase is the period of cytoplasmic growth. Mitosis is also divided into 4 stages, namely prophase, metaphase, anaphase, and telophase. Chromosome condensation occurs during prophase. Simultaneously, the centrioles have moved to the opposite poles. The nuclear envelope and the nucleolus disappear, and the spindle fibers start appearing. Metaphase is marked by the alignment of the chromosome at equatorial plate. During anaphase, the centromeres divide and the chromatids start moving toward the two opposite poles. Once the chromatids reach the two poles, the chromosome's elongation starts. Nucleus and the nuclear membrane reappear. This stage is called the telophase. Nuclear division is then followed by the cytoplasmic division and is called cytokinesis. Mitosis thus is the equational division in which chromosome number of parent is conserved in the daughter cell. In contrast to mitosis, meiosis occurs in diploid cells which are destined to form gametes. It is called the reduction division since it reduces the chromosome number by half, making gametes. In sexual reproduction, when two gametes fuse, the chromosome number is restored to the value in the parent. Meiosis is divided into two phases, meiosis I and meiosis II. In the first meiotic division, the homologous chromosomes pair to form bivalents and undergo crossing over. Meiosis I has a long prophase, which is divided further into 5 phases. These are lactotin, zygotin, pakaitin, diplotin, and dikinesis. During metaphase I, the bivalents arrange on the equatorial plate. This is followed by the anaphase I, in which homologous chromosomes move to the opposite poles with both their chromatids. Each pole receives half the chromosome number of the parent cell. In telophase I, the nuclear membrane and nucleolus reappear. Meiosis II is similar to mitosis. During anaphase II, the sister chromatids separate. Thus, at the end of the meiosis, 4 haploid cells are formed. So this was the chapter, guys. I hope आपको समझ में आया होगा. मिलते हैं अगले ऑडियो बुक सीरीज में. आई होप इस रिवाइज में आपका हेल्प हुआ होगा. कुछ हेल्प हुआ होगा तो ज़रूर कमेंट में बताना. एंड आगे हम क्या कर सकते हैं चैनल पे वो भी बताना. टिल देन बी